Welcome back, everyone. Next, we have Clene Inc. It trades on the NASDAQ under the symbol CLNN and is a late clinical stage biopharmaceutical company focused on improving mitochondrial health and protecting neuronal function to treat neurodegenerative diseases, including amyotrophic lateral sclerosis, Parkinson's disease, and multiple sclerosis. Happy to welcome President and CEO Rob Etherington and the CFO Morgan Brown. Welcome to the conference, gentlemen. Nice to have you.
It's a pleasure to be here. Thank you for the invitation.
Floor is yours.
So Clene is a most unique company we're thrilled to present this morning. The forward-looking statements are on your screen. You've seen these before. I'll be making a number of forward-looking statements during the course of this next 20 minutes or so as we focus on improving the health of the mitochondria. Critical for you and I, everything we do with respect to our moving, walking, talking, eating, chewing, and breathing is key to mitochondrial health and is especially problematic if you or I are unfortunate enough to have a neurodegenerative disease. Indeed, the World Health Organization has predicted that the second most prevalent cause of death in the next 2 decades are presently neurodegenerative diseases. A therapeutic breakthrough is urgently needed. Clene is pioneering exactly what is needed in our view, that is a catalytic nanotherapeutic to treat neurodegenerative diseases. And what we're doing in particular is targeting the improvement of mitochondrial function via the nicotinamide adenine dinucleotide pathway. That's a big word. Let's talk about it in a slightly different way. The way you and I use energy is very connected to energy metabolism within the neuron, and everything we do to move and walk and talk and eat and chew and breathe is connected. Now, we have completed a number of studies over the last couple of years, 2 in ALS, I'll be speaking about them in a moment, 1 in MS, and then 2 target engagement studies called REPAIR, with a third expected to read out in a few months this summer. And so there's a growing body of clinical evidence across ALS and MS that support the potential of CLNN to treat neurodegenerative diseases. Now, oftentimes patents are relevant in a broad discussion. In our case, they're super relevant, but the discussion is short because we have 150 granted patents plus a number of trade secrets. What we do is unique and proprietary to Clene. We own all of the asset. This is the asset I'm holding here in my hand, CLNN. Patients are drinking it every morning. You can see a picture of it here on your left. This is a nanocrystal suspension. If I was blindfolded and took a glass of my water or a drink of our CLNN, I would not be able to tell the difference. And indeed, all of the data I'm about to show you is actually placebo, is purple-colored water. So what we're doing is orally administering this asset, or if I have a patient struggling with ALS through my feeding tube. And what we're doing is targeting energy metabolism. Now let's talk about that a little bit more. So in your brain and mind, there are quite a bit of neurons that are driving this choreography that enables us to, as I said a minute ago, move and walk and talk and eat and chew and breathe. And all of that process requires a lot of energy. In fact, our brain is only about 3 to 5% of our body weight, but it consumes about 1/4 of our daily energy requirements to basically do all these things that you and I take for granted, everything with respect to our movement that gives us life. And so what we're doing is enabling the energy to come into the mitochondria to enable the neuron to survive and to function. And as part of this process, any energy reactive catalytic system also causes some negative effects, in this case, an increase in reactive oxygen species, and we're reducing that negative effect. So let's use a bit of a metaphor. If you drive a combustible engine car that uses gas, then the gas is driving the pistons and the engine to move the wheels, and out the back end, out your exhaust is a complicated exhaust that is washed by a catalytic converter. So this is what we're really talking about. Our acid is enabling the neuron to work more effectively as it works to do its job. So with that mechanism, brief introduction, let's get straight into the clinical data. And before that, I want to talk about safety. Now, the FDA is quite appropriately animated about safety in all respects. And we have over 800 collective patient years of exposure to CMAU8, with long-term treatment now nearing 5.5 years, actually. And we have some side effects of nausea, headache, occasional diarrhea, but these are mild to moderate in severity, do not cause complicating implications, and we have no related SAEs. That is very important and unusual in drug development. We have not a single related serious adverse event across any of our clinical program. Furthermore, as you take our drug over time, if you did see this nausea, this headache, or this diarrhea, the incidence is decreasing. That's what no temporal association means. And we have not a single no adverse effect level finding across our program. And this includes not just our clinical program, but also to our EAP programs, that's expanded access protocol, where individuals with either ALS primarily, but also some MS individuals as well, can take our drug with compassionate use. So let's get right into the data. I'm going to focus first on ALS. We completed the rescue study in Australia. This was a 9-month study in Australia that we've continued in open label extension with patients on drugs still now. Boy, goodness, we started that program in 19, so 5+ years. And Healy also was started in 2019. This is a partnership with Harvard and 50+ centers of excellence across the country. And this was a 6-month study compared to 9 months in Healy's case. And in Rescue's case. And also, we continue to treat individuals in our Healy open label extension. Now, we missed the primary endpoint in both Rescue and in Healy. If we had achieved the primary endpoint, then our market cap would be at a very different place at the minute. However, there are 12 other assets, unfortunately, in ALS that have missed their primary endpoints in the last 2 years, including ours. In fact, there has unfortunately not been a single victory with the primary endpoint of any ALS study recently, dating back all the way to Amylyx, which is now unfortunately also withdrawn from the market. Even Biogen's tofersen, which the FDA approved, did not meet its primary endpoint. And I'm going to get to how they got approved in a minute, because we're following a similar path. But what you see here is other check marks, including survival, delayed time to clinical worsening, preserved function, and biomarkers. So let me come back to this with first some explanation of our clinical data. So though we missed the primary endpoint in Healy, we did achieve our time to event survival endpoint with a 94 and 94% achievement, a p-value 0.04. And PAV, just to explain that, is permanently assisted ventilation, which means that my lungs have become so compromised they can no longer power my breathing unless I have permanently assisted mechanical ventilation, forcing air into my lungs for 22 hours a day or more. And so these were the 2 endpoints that we were checking for survival. You can certainly appreciate if I don't have a machine breathing for me, I will pass away. That also is a survival indication. And that was the secondary for the 30 milligram dose. And we see, and this is data we just showed the FDA in late November, we see that continue robustly for 6 further months in open label extension, even after the placebo individuals, the gray line, converts to active drug. We stay, P04, 78% risk reduction on time to death. And we also see in the double-blind period, the exploratory endpoint of delayed time to clinical worsening. Now, clinical worsening in the case of ALS is defined as either I die, a pretty evident form of clinical worsening, or I need a tracheostomy cut into my neck to have mechanical ventilation. My lungs are too weak to power my breathing, so I need assisted breathing through a non-invasive but BiPAP, or I can no longer swallow food or water anymore because my throat, my mouth, my esophagus does not enable that movement down to my stomach. And so if that's the case, I need to get a gastrostomy tube into my stomach where I receive all my food and water. So those are very important steps that are evident key parts of clinical worsening, we saw 71% with a p-value of 0.1 in the in the rescue exploratory. And in the Healy exploratory, we saw a pretty similar number, 74% risk reduction with a p-value of 0.3. Now, I talked about biomarker a minute ago. So this is, you're looking at neurofilament, these little squiggly things here in purple. These are cytoskeletal proteins that provide the structure and support for the cell. They're highly specific for neurons. And in ALS and in other neurodegenerative diseases, the neuron is attacked and it leaves this cytoskeletal protein remainder behind, and I can measure it in the bloodstream. It's an example of a biomarker that suggests the neuron's been pummeled, and as it dies, it leaves evidence of its dying process in the bloodstream. And so high neurofilament levels are associated with axonal damage, axonal neuronal damage. And what's going on here is if I have high neurofilament in ALS, it predicts a greater risk of clinical progression. So if I was to go to the doctor today and have a blood work done and be told, uh-oh, you've got high neurofilament in your blood, then that is a very problematic thing, suggesting I need to hang on, batten down the hatches, and prepare for functional deficit. And so we really want to try to follow this neurofilament. And in Clene's case, we are the only phase 2 program to have seen a neurofilament reduction from the Healy program with a P of 0.4. And as we follow these individuals for a further year in open label extension, this is the green is active, the gray is the placebo, we see a further 70% improvement for a total net difference of 17% geometric mean ratio difference. And we stay significant over that time, despite the fact, you see here this gray turns to green. That's because 90% of the individuals on placebo, the minute they were given the opportunity to move to active drug, they moved. And so now we're comparing active versus active. And once we get to steady state, about 6 months in and beyond, we start seeing a reduction in neurofilament even in the placebo individuals who converted. Now, the agency wanted to understand neurofilament deeper, and so we have made a number of announcements of late, including showing the data that we showed the FDA. And what they wanted to understand is if we had evidence linking plasma neurofilament with AU8 clinical benefit. So let's talk about that by looking at time to death or permanently assisted ventilation at month 12. And what we see here is an 83% risk reduction. If we look at the top tertile of neurofilament compromise, in other words, at baseline, the worst neurofilament, the top third of worst neurofilament, we see a p-value of 0.3 with an 83% risk reduction in death, or PAV, again, permanently assisted ventilation, meaning that I need a machine to breathe for me. And if we look at the top half, that stays consistent, 84% risk reduction and a stronger p-value, 0.1. So evidence clearly that the greatest benefit in participants with CLNN are those that have the highest baseline neurofilament in the study. Now, let's just, independent of that, let's just look at what percentage of people declined in our week 24 6-month data in the Healy program. And that answer is 57%, nearly twice what happened in placebo. So we saw a nice percentage of folks with a neurofilament decline. And if we look at those decliners, anywhere from some decline to a lot of decline, we find that the time to death or PAV benefit in long-term survival is even stronger. In this case, highly significant p-value 0.001 and a 91% risk reduction. So if you're on our asset and you follow the patient for 12 months, and if their neurofilament is declining, there is a 91% risk reduction of a survival improvement that occurs. Now, independent of neurofilament, we also showed the FDA just everybody in our clinical trial population to see if there was evidence supporting long-term survival benefit, period. Again, independent of this adjudication or this assessment with neurofilament at baseline. And what we see here, when we use the most conservative statistical model possible called greedy nearest neighbor match, it's a complicated statistical modeling assessment that looks at covariate adjustments and propensity matching with natural history controls. Now, that's a lot that I just said, so let me just break that down for a sec. What we did is looked at PROACT, the ALS Natural History Consortium, and Answer ALS. These are three different databases of a number of individuals dating back many years that had ALS, and we understood what their natural history would be. That if I'm patient A, B, or C, and I match those people, and then I follow them, and they didn't have opportunity for drug, which, by the way, unfortunately is tragic. Most patients still have the only option for riluzole, which was approved in 1995, 30 years ago. That is the primary drug still approved and prescribed in America and through the rest of the world. So in other words, there is a desperate need for new drug development in ALS. But in any event, if I look at the natural history of individuals against what we are actually seeing, when I look at this over 3 years, I see a 36% risk reduction across Healy alone and across Healy and Rescue taken collectively. The p-values of 0.2 and 0.1, suggesting very clearly that there's an improved long-term survival against where I would have been if I was looking at natural history. And now taking this even further to look at real-world evidence, and by this I mean individuals that are such struggling with their ALS that they could not otherwise have an opportunity to be treated in these clinical programs because they were too ill. We've offered the option for many of these individuals, actually nearly numbering 500 at the minute, into our compassionate use program. And we track those individuals, and we have 4-year data here because we've been offering compassionate use EAP drug to individuals since 2019. So that's why this data is lengthy in its time course. We see a 31%, again, same concordant type of improvement with the same strong p-value of a 0.06. Now, interestingly, Clene just presented a month ago, and we are presenting this further at an upcoming scientific meeting in ALS, data that shows against regimen A concurrent controls. Now, just a little bit of explanation what I mean by regimen A. In the Healy program, originally there was 3 programs that enrolled, regimen A, B, and C. Clene was C. 2 other companies were regimen A and B. And all 3 of those programs included all patients that met the same inclusion and exclusion criteria. They were effectively matched. And once matched, they were randomized into either A, B, or C. So we had no idea, nor did B or A, which patients we got. Each one of us got 120 actives and 40 placebos. And so those 160 patients per A, B, and C were all started at the same rough time, enrolled at the same beginning at the same time, and followed for the same period, 6 months, using the same criteria and the same endpoints. And regimen A, unfortunately, was stopped for futility. It became very evident that there was no benefit of the drug. And so everybody was stopped, and those individuals were rolled over into other programs, either other compassionate use programs or others OLE. But what we did is we were very grateful because the sponsor of regimen A provided this data to us in its entirety. And we used that data as what's called a concurrent control. So they weren't classically placebo data, right? Because they weren't our placebo, but they were the same patients that never had access to our asset and were randomized in the same fashion. And we saw that if we compare against those, we see, again, around a 30%, in this case, 28% risk reduction with a p-value that is, again, significant if we look at the full randomized set. So what it does is the Healy program was not run for that long, 6 months, and was not run for that many people relatively. But we do find, as we continue to follow these individuals for now up to 3 years in this data, that we see a consistent and concordant survival benefit that is similar to what I've already showed you in other analyses. And what's especially intriguing to us is as we talk about the FDA path going forward, the FDA does expect that if the accelerated approval path works that we're presently planning for, we need to nonetheless start a phase 3. We're planning to start such phase 3 by the end of the year. And we see a 50% risk reduction in this case. So now I want to just jump into MS for a minute, because I just presented very key data at the American Academy of Neurology only a week ago. This is a study looking at vision and function. And what we saw in that study when we first announced it, that AU8 at the same dose, 30 milligram, was improving function and was improving vision. The primary was vision, the secondary was function. As we continue to follow these patients, we see their vision improves quite appreciably. This is 8 points. So what does that mean? Here's the eye chart. That means 8 further letters I'm able to improve. And placebo people, as you see, they also, once they move to active drug, improve. We saw that cognition improves as well. This is a very important piece. Cognition is also improving over time. This remains one of the key debilitating elements of MS. And why is this occurring? We just showed data that suggests that the MRI and the VEP measures, which are paraclinical biomarkers looking inside the brain and looking in the eye, we are improving latency, amplitude, which is the amount of and the speed of the information that my eye is processing, which is why I can see better. And we're also improving diffusion tensor imaging, which shows just better whole brain white matter within the brain. And it's statistically significant, and it is actually beautifully correlated. So what's next? So as we close here, we are taking all this data to the FDA in 2 different engagements. So let's talk about MS briefly, because that's a little further off. So we're organizing an end of phase 2 meeting with the FDA, and we'll be talking to them about what could be a categorically market-leading opportunity in MS. Presently, MS people there's lots of them. There's a lot of billions of dollars spent on MS drugs. The drugs are disease-modifying drugs, so they're tamping down the immune system to prevent further MS attacks. But what these drugs do not do is they do not improve function. They do not improve vision. They do not improve cognition. And patients, as I said already, are already complaining of a consistent MS cognitive compromise. The MS patients call it cog fog, cognitive fog. My mind doesn't think clearly. There's a high amount of disability because of this. And the DMTs that are presently available don't provide improvement there. Our asset appears to be doing that because it's protecting the neuron and remyelinating. So we're talking to the FDA about this for the possibility of the start of a phase 3 study, even whilst we also prepare for the repair program and progressive MS to read out later this summer. Now, this probably requires a partner. MS programs are expensive, so we're talking to partners presently. Now, on the ALS side, different story. Because of the neurofilament benefit and the survival benefit, the possibility exists for possibly an accelerated approval pathway. We'll find out in the summer if our neurofilament from our expanded use that the NIH funded, $45 million of funding, I should add, where neurofilament is being checked every 6 to 12 months, if that data is concordant with the rest of the neurofilament data, and if it is, we're into the agency with the possibility of a new drug application in the fall. So, closing here for some questions. AU8 is the world's first gold nanocrystal suspension and in development, as I've shown you, to remyelinate and protect neuron function. A lot of data in MS and ALS, well more than 800 patient years of safety. So safety becomes not a dispute. We own the asset outright, so any potential partner has the ability to come in and work with Clene directly. And then we have cash on hand to get us to the end of these FDA periods as we conclude the neurofilament data set and run back to the FDA with a planned meeting, as we've announced it, that's scheduled and which we're preparing for now later this quarter to talk about the neurofilament assessment and the possibility of this new drug application. So that was 24 minutes of a lot of data. Let's have some questions.
Great job. And thank you, Rob. I know you just shared this in your presentation, but can you please reiterate what your upcoming milestones and maybe inflection points are and when they might occur?
Sure. So first of all, I have a presentation of some more ALS survival data coming up at a key scientific meeting in June. Also, at the end of this quarter, I'm expecting an FDA meeting to discuss our statistical analysis plan. That will then lead to the key data. This is the most important catalyst is the look at the neurofilament from our NIH-sponsored EAP program with 180 subjects, which is, just to be clear, 3 times the number of ALS neurofilament data that I got from Healy. And if that data is concordant, then the agency expressed to us, and we've announced this in a press release in December, the possibility of an NDA filing. So once we get that NDA filing, that becomes another catalyst. That would happen in the 3rd quarter, and then that acceptance of said NDA filing would be another key catalyst in the 4th quarter.
Perfect. Talk a little bit more about that NDA. Are there any comparators as to what your stock price might do should your NFL data being concordant with your clinical trial data and you file an NDA that is accepted by the FDA? So what then?
So it's a little hard to always predict stock price, especially in the present kind of very complex markets that we have at the moment. But if I use a surrogate, there's another ALS company that did have their drug on the market for about a year before they had some unfortunate news and had to pull their drug off the market. But in that case, they were trading, kind of in a complex way. It was a different market then, so it's hard to make a complete comparison. But let me say, when they got their NDA approved and rather accepted and then approved, they went in the end to $1 to $2 billion they were planning to sell up to $400 million of their ALS asset to a desperately needy cohort of ALS individuals who are struggling with this disease. So now the patients, as I said a moment ago, have really effectively only riluzole or maybe edaravone uniquely in this U.S. country. There's a desperate market need for a therapy that can remyelinate and protect the neuron and provide the energy it requires. And so we do believe, actually, that if the FDA approves our asset, there is a very large opportunity in the hundreds of millions per annum in terms of potential revenue.
Perfect. And share your plans with the P3 trial in ALS, and when will that start?
The plan there is to start that study because the FDA requires an accelerated approval, that there is an ongoing study. That is a necessary component of underway. They use the word underway when you consider approval. It has to be underway by the time of approval. So if we're talking an NDA submission in the fall, and that approval could be sometime next year, then we're planning an underway in the second half of this year, first only in Australia, and then more broadly once we understand the FDA path next year. Such a phase 3 study is very expensive, and so we are starting only in Australia while we do this first filing period to know definitively how we plan to work on approval and commercialization next year. And by the way, that phase 3 study, I should add, critically, is all around survival. The endpoint is time to event survival. We think this is the critical need in ALS, that patients are not passing away, that they are living.
And Ed wants you to clarify, this is 150 patients or 150 patents?
150 patents. So Clene has 150 patents. Wow. If I said patients, forgive me. If Clene has 150 patents, so we're very effectively patent protected through the globe. Those 150 patents are on our nanotherapeutic technology process, method of use, and around the globe in multiple all the key pharmaceutical countries.
Very impressive. So are they all related to ALS and MS? And have you any products in the market currently?
We don't have any markets in the country currently. This lead asset, CNM-Au8, is our lead asset. And yes, those patents are related to MS, ALS, and Parkinson's disease, both method of use, which means this target to these diseases, and also the way we do this. There's nobody else in the world that prepares a clean surface, that is to say, without any chemistry, a nanocrystal suspension as a drug. Nobody else does this. We've been doing this 10 years. There's not been a single competitor arise because what we do is most unique, most proprietary, and that's the reason we have a lot of trade secrets also.
And Eli asks if you can talk about what you are facing in the regulatory environment.
The key question there is, the FDA is under, and we read the headlines every day like you do, the FDA is under the middle of a key transition. And we'll see how that landscape lays out. But the direction that we were given in December, and we announced this publicly, is to come back and speak to us about your more robust neurofilament data that's emerging. And then if that is concordant and you can leverage the persuasiveness of that argument, then there's the possibility of accelerated approval path. And so, there's a new leadership team in play. Will that be more supportive to Clene? Possibly. Will that be, in particular, there's some individuals that have now been moved up the ladder to high ranks that understand nanotechnology more so than previously the FDA had in place. And also the team that we are still meeting with are still there. The same folks that gave us this path in the neural division that we're working with are still presently at the agency. And as I said a minute ago, we have a meeting with them coming up later this quarter.
Perfect. Well, Rob, thank you for this very thorough and very important presentation. We appreciate you being on the Emerging Growth, and we would love to follow along with some updates in the future.
Thank you so much. It's my pleasure, and I appreciate the time.
All right. Thank you, Morgan. Thank you, Rob. We'll see you again soon, and we'll be back with our next presenter.